Okay, we are starting on slide number 7 of the chapter 21B PowerPoint slides. So antigens can be complete or incomplete. So an incomplete antigen is called a hapten. If they're complete, then they have two important properties. Immunogenicity, which is the ability to stimulate the proliferation of specific lymphocytes. In other words, complete antigens will stimulate lymphocytes like T cells and B cells to clone themselves, to grow, to divide. And reactivity. Complete antigens have reactivity. They have the ability to react with activated lymphocytes and antibodies released by immunogenic reactions. So they can react with our immune system. Examples of complete antigens are foreign proteins, polysaccharides, lipids, and nucleic acids. These can be seen on many foreign invaders such as pollen and microorganisms like bacteria. Now, an incomplete antigen, just so you know, is called a hapten, and sometimes they don't, it takes a little bit more to cause them to be reactive with our immune system, okay? So, So sometimes a hapten can cause the immune system, which is just an incomplete antigen, can cause the immune system to mount an attack that's harmful to the person because it attacks self-proteins as well as the hapten. Examples are poison ivy, animal dander, detergents, and cosmetics that cause allergic reactions. An antigenic determinant is a small part of an antigen. It's the part that binds to the antibody. So if this is your antigen, the antigenic determinant is this piece here, and this piece, and this piece. Okay? And the Y-shaped blue molecules are antibodies that are binding to the antigenic determinants on the antigen. Okay? Now, a self-antigen, of course, is recognized by our immune system as a self-antigen so that our immune system won't attack it unless you have an autoimmune disorder. One group of self-proteins or self-antigens are called MHC proteins, which means major histocompatibility complex, and that means tissue compatibility. And these are unique to each person. All right, section 4, we're going to talk about the adaptive immune system and lymphocytes and APCs, which are antigen-presenting cells. These are cells that will present the antigen on their surface. So, the adaptive immune system involves 3 crucial types of cells. There are 2 that are called lymphocytes, and they are B cells and T cells. B cells are responsible for humoral immunity, and T cells are responsible for cellular immunity. Antigen-presenting cells don't respond to specific antigens, but they do present the antigen on their surface after they've engulfed the antigen. So, lymphocyte development, maturation, and activation. All right, so first of all, both B and T cells originate in the bone marrow, but B cells mature in the bone marrow, whereas T cells mature in the thymus. So in the maturation process, lymphocytes are, quote, educated. This takes 2 to 3 days, and it happens in the primary lymphoid organs. So B cells are, they mature in the bone marrow, and the T cells mature in the thymus. This should say T cells in thymus. Let me fix that. Sorry. Okay. Now, the reasons lymphocytes are educated is so that they can recognize only one specific antigen. And the second reason is for self-tolerance, is so that they won't respond to self-antigens. Then you have antigen encounter and the activation of the lymphocytes. So the first encounter with the antigen activates the lymphocyte, and it causes the lymphocyte to begin to divide, and this is called clonal selection. And then proliferation and differentiation means that they're gonna divide and grow in number, and then they're gonna differentiate into specific types of T and B cells. So most of the clones of B cells and T cells become effector cells that actually fight infections. Some of them remain as memory cells that can fight the same infection if it comes back a second time, and that the memory cells can mount a very fast immune response to a second attack. And then these effector cells and memory cells circulate continuously. Okay, so the education part for T cells, again, they mature in the thymus. They're formed in the bone marrow, but they mature in the thymus. And with positive selection, this selects T cells that are capable of recognizing self-MHC proteins. Those that can't are destroyed. So positive selection selects T cells that can recognize self-antigens, and destroys ones that can't, because the ones that can't are going to attack them. And negative selection prompts T cells that bind to self-antigens to undergo apoptosis. And this is called clonal deletion, because we don't want them attacking self-antigens. And this shows you the positive selection. T cells must recognize self-MHC proteins by binding to them. If they don't bind, that results in death. If they do, then they survive. And then move on to negative selection. The ones that recognize and bind to a self-antigen then undergo apoptosis, and the ones that don't survive. Because if they ever bind to a self-antigen, then that's going to mount an immune response against it. Okay, so antigen-presenting cells are the third type of cell that we're going to talk about, and APCs engulf antigens and present those antigens to T cells for recognition. The three major types of APCs are dendritic cells, which you find in your lower layer of your skin, macrophages, and B cells. So all of these can actually engulf antigens and then present fragments of those antigens on their surface to T cells, so the T cells can recognize those antigens. Dendritic cells are found in connective tissues and the epidermis. They phagocytize pathogens that enter tissues, and then they present the antigens on their surface so that T cells can bind to them. This happens in the lymph nodes. Dendritic cells are the most effective APC known, and they are a key link between the innate and adaptive immune system. There's a picture. They have all these extensions that can reach out and grab harmful pathogens and engulf them. Macrophages are the large white blood cells that are part of our innate immune system. They are also known as big eaters. So they're widely distributed in connective tissue and lymphoid organs like your spleen. And they present antigens to T cells. This is going to activate the T cell and further activate the macrophage, which will then become a phagocyte, phagocytic, to those antigens when it encounters them. All right, the humoral immune response is controlled by B cells. B cells produce antibodies that attack antigens. So most cloned B cells become plasma cells, and plasma cells are specifically the type of B cells that secrete antibody. And antibodies can circulate in the blood or the lymph, and they will bind to specific antigens and mark them for destruction. The B cells that do not become plasma cells will remain as memory cells. In the primary immune response, when the immune system is attacked by a specific antigen for the first time, there's a 3 to 6 day lag period. By 10 days, you have your blood will have reached peak levels of antibodies in the blood plasma. And then after 10 days, you will see those levels of antibodies decline. In the secondary immune response, which is when you're exposed to the same antigen a second time, so a second attack, you have memory cells that provide immunological memory, and they're able to respond to that particular antigen within hours, not days. The antibody levels don't take 10 days to peak, they only take 2 to 3 days. And they bind with greater affinity, so the binding is a lot stronger. All right, we have two types of humoral immunity. There's active and passive. Active occurs when B cells encounter antigens and produce antibodies against them. So there's naturally acquired, that is when you actually get sick from something, and then you're immune to it after that first sickness. And then there's artificially acquired, which is when we get a vaccine. Passive immunity is when you receive ready-made antibodies. This is passive. It means your immune system is not making its own. You receive ready-made antibodies. So you can get these naturally through the mother to the baby or through breast milk. Or you can get these artificially through an injection of antibodies, such as monoclonal antibody injections, like they gave some people who had COVID. There are different reasons for monoclonal antibody injections, but when you are given injections of antibodies, that is artificially acquired passive immunity. So make sure you know examples of the 4 different types of humoral immunity: naturally acquired active, artificially acquired active, naturally acquired passive, and artificially acquired passive. As far as antibodies, they also can be called immunoglobulins, or even, what was the other term? I can't think of it. We said that let me see if I see it. Agglutinin, okay. Antibodies are agglutinins and antigens are agglutinogens. But anyway, we're just going to call them antibodies or immunoglobulins here. But they are secreted by plasma cells, which of course began as B cells. And there are 5 classes of antibodies. There's IgM, immunoglobulin M, IgA, IgD, IgG, and IgE. So there's 5 classes, and you just need to know examples of each of the 5 classes. IgA is found in mucus and other secretions like tears. IgD functions as a B cell receptor, and you see IgD antibodies attached to the surface of B cells. IgG antibodies represent 75 to 85% of the antibodies in our blood plasma. They can cross the placental barrier from mother to fetus. And IgE antibodies are active in some allergies and parasitic infections and cause mast cells and basophils to release histamine. And then you have a table that shows you the IgM, IgA, IgD, and IgE antibodies or immunoglobulins.